Hello, everyone. Good morning. I think we should start. Welcome to Open Box Science. Today's presentation will last about 40 minutes, followed by a 15 minutes question answer part. And then today's presentation title is Function and Regulation of Nucleotide Variant in RNA. And the speaker today is Giovanni. Giovanni obtained his bachelor degree from the University of Florida and PhD from UCLA. Currently, he's a postdoc. He is a postdoc at UCLA and investigating the regulation of RNA processing use third generation sequencing data. Here is a fun fact about him. His very first research related task was to water plant. Oh, sorry.
Right.
Hi, Joan. Feel free to take control.
Yeah. Thank you so much for the introduction. Today I'll talk about the function and regulation of nucleotide variants in RNA. And this sort of captures the two projects that I'll dive into a little bit more detail. So firstly, when we talk about nucleotide variants in RNA, thing where most of us are probably familiar with the process of transcription, which is the process by which we make an RNA copy of DNA sequences of a gene. And any genetic variant that is present at the DNA level will then also be present in the RNA sequence. And of course, potentially, these mutations could also affect the final protein product through the process of translation when we convert RNA into protein. Now, these genetic variants, we can categorize small genetic variants into three categories: SNPs, insertions, and deletions. And this type of variants have been very widely studied. Many different types of analysis have looked into the role that this type of genetic variants play in different traits, in different diseases, and so on. A lot of times focusing only at the DNA level, but also trying to focus on the effect they may have at the level of RNA. And to further increase the complexity of this, we also have a second class of variants that are present in the RNA, which here we call post-transcriptional modifications. And there is a number of RNA modifications that have been reported, but one of the most common ones, pretty much, well, depending on who you ask, depending on definition, one of the most common ones is RNA editing, which is the process by which you modify an adenine base into an inosine. And these RNA modifications are not present at the DNA level, but will be present in your RNA transcripts and can potentially also affect the downstream phenotype, or in some cases, the final protein product. So when we talk about nucleotide variants in RNA, this concept captures both genetic variants that are expressed in RNA transcripts and post-transcriptional RNA modifications, such as RNA editing. Now, this RNA editing is a process that happens fairly regularly within the cell. Today, when looking at the different databases like Reddit or Radar, there is about 4.5 million H2I editing sites that have been reported. So to sort of summarize the flow of the talk today, I'm looking at this this process by which we have different RBPs or RNA-binding proteins that will regulate these different nucleotide variants, in this case, the nucleotide modifications. And once we have these nucleotide modifications or other genetic variants in the RNA, we're also going to see how we can detect them and how we can potentially assign them a function that will tell us something about the final RNA transcripts. So the first question is, what are the factors and mechanisms that are involved in regulating RNA editing? Second, when we come to detection, we also want to ask, how can we confidently detect variants at the RNA level? And how can we find a systematic way to characterize them, to learn a little bit about these variants and what is their role, at least at the transcriptome level? To address these questions, I'm going to talk about these two works. The first one that looks at the regulation of RNA editing by RNA-binding proteins, and the second one that talks about this new method that we published called scAllele, which deals with the identification and functional analysis of variants. So diving a little bit into the first paper, the regulation of RNA editing by RNA-binding proteins, I wanted to first introduce this concept of RNA editing, adenosine to inosine editing more specifically. And this is the process by which an adenosine base is deaminated. So this amino group is removed and converted into a new base called an inosine. Now, this process is carried out by a family of proteins called ADAR. And the resulting nucleotide, this inosine, is read by the different protein machinery, say like the ribosome or other RNA binding proteins, as a different base. So the canonical adenosine pairs with a uridine at the RNA level, but the converted inosine will pair with a cytidine. So in terms of base pairing, this inosine behaves very much like a guanine. This process of RNA editing is very highly regulated. So say individuals are from different ages will have different editing levels. When your body is undergoing an immune response, for example, your RNA editing levels are also going to be different. We also observe that this regulation of A2I editing is observed in multiple diseases. So it's been reported that certain types of cancers will have higher RNA editing levels, whereas also in many neurological diseases, it's also been reported that the RNA editing levels is very different from that of normal. So it is a very tightly regulated process. And what we observe is that although the ADAR proteins are the ones carrying out this modification, it's not enough to look at the expression of ADAR to figure out what are the overall editing levels present. It's being long recognized that there are multiple factors that regulate this process, but it has not been very systematically studied to figure out what are those factors and how exactly are they influencing RNA editing. So, of course, a natural candidate to look into are RNA-binding proteins called RBPs. RBPs in general, they regulate multiple processes, multiple steps of RNA processing. And there's been reports of multiple RNA-binding proteins that directly interact with ADAR. So making use of ENCODE, some data sets put together by the ENCODE consortium. More specifically, we have RNA sequencing corresponding to the knockdown of over 200 RBPs. So this type of data allows us to study the effect of every RBP on the overall RNA editing level. We identify and quantify RNA editing sites. That is by looking at the RNA sequencing data, we find specific positions in the transcriptome that are edited. And we also obtain an RNA editing level, which is how many reads that map to this position are edited versus how many are not. And we also perform differential RNA editing analysis between RBP knockdown and control. And this is how we can quantify the effect of that one RBP on the overall RNA editing levels. This figure is very complex, but it does very nicely summarize our results. This is results from one of the two cell lines that we studied, K562. And what this figure is showing, we have bars for all the RBPs that we looked into. The color of the bars indicate the overall direction of editing change. So for example, bars that are more in yellow that have a negative value, these are RBPs that when you knock them out, you have lower RNA editing than compared to the control. So of course, we have our first candidate, which is ADAR, which is the actual RBP that carries out the editing, having the strongest effect. But you also find other RBPs that have a similar effect when you knock them down, such as DROSHA and FXR1. You also have RBPs that when you knock them down, you observe the opposite effect. That is the example with PUS1, for example. This is a pseudouridine synthase. When you knock PUS1 down, you observe increased RNA editing level. The height of the bar indicates the fraction of differentially edited sites. That means out of all the testable RNA editing sites, how many of them are differentially edited compared to control. And the links between them is a measure of overlap, pretty much how many shared differentially edited RNA sites in the same direction of editing between two RBPs. So the thicker the link, that means the more RNA editing sites are commonly regulated by this pair of RBPs in the same direction. So we're going to dive a little bit into some examples and some mechanisms that involve this regulation by RBPs. The first thing we did, however, is to first corroborate our results. The amount of regulation that we report is based off the number of differentially edited sites. But we also wanted to see if we can explain the observed average editing levels based off only the expression of the most significant RBPs. So for the different RNA sequencing samples that we had, we calculated the RBP expression in terms of RPKM for the most important regulators and try to see if they would predict the overall observed RNA editing level. And as you can see for this cell line, we observe a significant correlation between the expression of the top 15 most significant RBPs and the observed editing level. We wanted to figure out exactly how is this regulation taking place. So one of the first candidates that we looked into is this protein called TDP43, which interestingly enough is both an RNA and a DNA binding protein. And when we look into, first of all, when we look into what of these RBP knockdowns have the greatest effect on either one expression, we plot in this plot over here, you can see we have the ADAR1 RPKM in the x-axis and the log2 fold change in the y-axis. So you can see the expression of the ADAR protein in the different knockdowns, RNA-seq knockdowns that we have. So for example, when we knock down ADAR1, then the ADAR1 knockdown is going to have the lowest ADAR1 expression. And this is true for both cell lines. But we also observed that when we knock down TAR-DBP, this is the gene that codes for TDP43. When we knock down TAR-DBP, we also observe a very significant change in ADAR expression. And as you can see by the histogram on the y-axis, you can see that it's a overall, based on the other knockdowns, this RBP has a very significant effect on the expression of ADAR. So naturally, we assume that TDP43 is acting as a regulator of RNA editing at the level of ADAR expression, so very upstream. So thanks to the help of two of my lab mates, Dr. Jay Bahn and Dr. Hyunik Jun, they performed this Western blot where we looked at the expression of ADAR1 in the two cell lines and under different conditions. We have control cells, and we also have two duplicates of cells with TAR-DBP knockdown. And we can see that in the HepG2 cell lines, when you do knock out TAR-DBP, you observe very reduced expression of ADAR1, something that is not observed in K562. So that also explains why we observe TAR-DBP as a very significant regulator in the HepG2 cells compared to the K562. And of course, when we look at the overall changes in RNA editing for the TDP43 knockdown, we see these directional changes where the control samples, so the samples that do have TDP43, have higher editing levels compared to the knockdown. And the numbers here show the number of differentially edited sites above and below the diagonal line. So sort of expressing this, the direction of RNA editing. So when we look into other types of data set that can give us some information about this type, about how exactly TAR-DBP is affecting the expression of ADAR, we looked into ChIP-seq pretty much binding sites at the DNA level of TDP43. And we find something very interesting. We find that there are significant binding regions of TDP43 that overlap with transcription start sites of ADAR protein. So when we show when we overlay the ChIP-seq peaks with the ADAR transcripts, we can very clearly see that there is one peak that is consistent in 2 replicates that overlaps the transcription start site of the P110 isoform of ADAR. And there is one peak that appears in one replicate that overlaps the transcription start site of the P150 isoform of ADAR. So this and these peaks also overlap other significant features. For example, we have other regions of DNAs that were found from DNase assays that show regions of open chromatin. We also have regions of polymerase II binding and other histone marks that sort of all highlight these regions as regions of open chromatin transcription, and for the ADAR1 gene where TDP43 is binding. And to further validate this mechanism, Dr. J. Ben and Dr. Hyunick Jun also perform different reporter assays where we introduce the sequence of the peaks that we found from the TDP43 ChIP-seq into reporter assays containing enhancer promoter sequences. And we see that every time we introduce the sequence of the peaks that were identified, we observe differential expression of the increased expression of the reporter gene. So confirming that these regions are in fact affecting the overall expression of the downstream ADAR gene. Next, we also, another category we wanted to dive in is to look at RBPs that have been reported to interact with ADAR. So in the literature, there's been a number of reports about different RNA-binding proteins that interact with ADAR with or without the help of RNA. And of course, if an RBP has a direct interaction with ADAR, then it's naturally to wonder if they're also having a role in RNA editing. From our data set, we identified 18 RBPs that were ADAR interacting. And from those 18, 8 of them had very significant changes in RNA editing. So this is a similar plot as before, where we see the bias in the editing ratio in control versus knockdown. And here we have ADAR1 as a reference for the 2 cell lines. And we report a number of, and these are all ADAR interacting RBPs. And we observe a number of them that have a very significant effect of editing in the same direction as ADAR. So I previously mentioned DROSHA and FXR1 from our first figure, how they have this very upregulating bias so that the control has higher editing levels than the knockdown. Similarly, we also find ADAR interacting RBPs that have the opposite direction. So this downregulating bias, where the RNA editing is higher in the knockdown. And here we have other RBPs such as ILF2, PAB, PC1. So we already know from previous assays that these RBPs interact with ADAR, but then that still doesn't tell us whether they are binding to the same regions as ADAR, more precisely to the regions that are getting edited. So we make use of another type of data set, which is called ECLIPSE-seq. ECLIPSE-seq gives you information about the binding sites of in RNA of different RNA binding proteins. So what we did is to find the distance of the closest binding site of each RBP to each of their differentially edited sites. So once again, we put here ADAR1 as a reference. So you can see that ADAR1, if you look at the orange curve, it binds very closely to the differentially edited sites. And here we have these gray curves that serve as controls when we select non-differentially edited sites or random A bases within the transcriptome and measure the distance to those as a control. So we observed that ADAR1, of course, binds very close, as expected, to the RNA differentially edited sites. But then there are other proteins that stand out, like DROSHA. So we already know it has a very strong regulating effect. We know it interacts with ADAR. And now we also know that DROSHA binds very closely to the differentially edited sites, which suggests that the type of regulation it has on RNA editing is very local, as opposed to, for example, TDP43, where we just saw that the type of regulation is by affecting the expression of ADAR. Other ADAR interacting RBPs like FXR1 and MADAR3 don't have a significantly closer binding pattern. So for these RBPs, we cannot say they have a local type of mechanism, and they may affect RNA editing in a more indirect way. And similar to DROSHA, we also have another RBP, XRCC6, which, although the signal is not as strong, it still has a significantly closer binding position, suggesting that it could also be could also have a local effect on RNA editing. One RBP that stood out from this analysis is ILF3. And here we show the same plot for this protein, for this RBP in the 2 cell lines. And what we observe is that the binding profile of ILF3 is very similar to that of ADAR. So if you remember, ADAR has this very narrow peak. If we look at the metrics, if we look at, for example, the median of the distance to the differentially edited site, about 0.5, 0.3 in the 2 cell lines. And DROSHA, that was very significant, is about 1.4. When we look at the binding profile of ILF3, we even see a stronger signal than that of ADAR, with a median of 0.3 and 0.1 in the 2 cell lines. So this binding profile tells us a number of things. For example, if we look at the overall effect of ILF3, we see that it's not the strongest regulator. It's not the RBP that has the highest effect on global RNA editing. But at the same time, it is an RBP that is binding very closely, almost a perfect overlap with every differentially edited site. So what this data is telling us is that this RBP it does interact with ADAR based on previous assay, and it is also binding very closely to the differentially edited sites. It has a very selective mode of regulation. It is binding to the regions that are edited, but is not strongly regulating every time. And this observation is also reported in other studies where ILF3 is found as a modulator of RNA editing in cancer. And when we look at the motifs or the regions in the transcriptome where ILF3 is bound, we see that ILF3 is the RBP that most strongly binds to Alu elements. Now, Alu elements is a type of repeat that is present in the human genome. And the thing about Alus is that they can form these double-stranded structures and double-stranded structures are the structural motif of the ADAR proteins. So regions in the transcriptome that form double strands are very likely to get edited. And when we look at the profile of, say, ADAR, once again for reference, we see that around half of the time it binds to the sense direction of Alus, and the other half to the anti-sense. So this even split, what is telling us is that the sense, of course, the sense sequence of an Alu is going to be different from the anti-sense. And if you are binding to either one of these two sequences, is that it's not the sequence that you are targeting, is the structure, is the double-stranded structure. And when we look at the different RBPs that also bind to Alu elements, we also find ILF3 that have a similar pattern where it binds around half of the time to sense and half of the time to anti-sense Alus in the two cell lines. So similarly, this also suggests that ILF3 is binding to these double-stranded regions. That also explains the proximity to the binding of the differentially edited sites. Other RBPs, for example, HNR-MPC, that very frequently binds to Alu elements, but we observe that 97% of the time it binds to the anti-sense Alu. That means that shows a strong preference for the sequence of the anti-sense. So this would suggest that proteins like HNR-MPC have a stronger binding motif rather than structure. Now, as I mentioned before, double-stranded structures mainly formed by inverted Alu sequences, those are the primary targets for RNA editing. So the question is, do RBPs that frequently bind to Alus, are they strong regulators of RNA editing? What we find is that when you look at the fraction of ECLI peaks or binding sites that overlap Alus, and the fraction of sites that are differentially edited, we actually see a very poor, almost non-correlation. You see that ADAR, all the way up here in the top right corner, has a high fraction of binding sites overlapping Alus, and a high fraction of sites that are differentially edited upon knockdown. But we don't really observe an overall pattern in the set of Alu-binding RBPs in our data set. We do observe that some RBPs that overlap Alus very frequently have a smaller effect on overall RNA editing levels. We also observe another RBP that binds to Alus around 12% of the time, meaning around 12% of the binding sites overlap Alu elements, and yet have a very strong effect on overall RNA editing. So this observation, of course, suggests that not binding to the Alu elements is not enough to regulate RNA editing. And this is also supported by the fact that RNA editing could happen at multiple steps in the processing of RNA. It could happen very early in transcription. It could happen also when the transcript is already in the cytoplasm. So this long span of time where RNA editing could happen, the sequences, the elements, the Alu elements could have been bound by a number of different RBPs. And of course, not all of them will have an overall influence in the editing process. An interesting sample that we find is TROV2, which is one of the RBPs that we looked at and that is present in, is characterized by downregulation of RNA editing. And what we find is that this is also reported in the literature in patients of systemic lupus erythematosus, where these patients, they are characterized by production of anti-RO60 antibodies. RO60 is a protein encoded by TROV2. So we observe in these patients that there is also a similar editing pattern than that of the TROV2 knockdown. However, these patients also have differential ADAR expression. So we have two factors pretty much explaining the difference in RNA editing. We have differential expression of RO60, and we also have differential expression of the ADAR proteins. So we aim to see if RO60 alone can explain that difference in editing. And although we already have the knockdown data, we also try to validate those findings by looking at a different data set that had TROV2 overexpression. And what happens is that when we overexpress TROV2, we observe the opposite pattern where we have downregulation of RNA editing sites. And in this overexpression of TROV2, we see no difference in the expression of any of the ADAR proteins, suggesting that TROV2 alone or RO60 has that effect in mediating RNA editing and is a factor that explains differential RNA editing in SLE patients. So as a summary, we surveyed over 200 RBPs for potential RNA editing regulators. We highlight a number of RBPs and their mechanism, such as DROSHA, RO60, TDP43, ILF2 and 3. And we also highlight some mechanisms of regulation that including the modulation of ADAR expression, ADAR binding, and Alu-binding. I do have some slides for the second project, but I think we probably don't have time for that, which is our tool scAllele. I'm happy to talk about it if anybody has questions, but I think for now, I'm just going to skip to my last slide. Where I wanted to say just thank you to members of the Xiao lab, which is my lab at UCLA, And also open box science, Dr. Quan Lin, Dr. Xishen Wang, for giving me the opportunity to talk to you, to every one of you today.
Okay, thanks for Giovanni's nice presentation. Anyone have any questions? If you have a question, feel free to ask unmute and ask directly.
Giovanni, I wonder if you can present on the single cell allele part for 5 minutes, because I think some audience might be interested, or 10 minutes.
Oh, yeah, for sure. Yeah, let me just go back. Okay, yeah, it's gonna be a challenge. Let me see if I can wrap it up in 5 minutes. Gonna skip over some of the motivation about making a variant color. This slide summarizes the project. I think the motivation. We want to make a variant color plus variant analysis tool that calls nucleotide variants in single cell RNA sequencing using local reassembly. One of the challenges of single cell, of course, is regions that are very lowly covered. So we want to make it very sensitive to low read coverage. And at the same time, after identifying variants, we want to make some functional analysis. So we use this feature of real-level variant calling to find linkage between variant alleles and splicing isoforms.